Okay, so to draw DNA structure, I'm redoing what I did about a month ago because what I did about a month or 2 months ago was a mess. So you got basically, that's adenine, and then you got now here comes the fun part. They put the hydrogen bonds in. That's oxygen to hydrogen, nitrogen to hydrogen. Oxygen, nitrogen, and nitrogen are higher electronegativity than hydrogen, which is why you get hydrogen bonds. They pull the electrons, they attract electrons from the hydrogen to them. Okay. So this becomes slightly negatively charged, and these become slightly positively charged. So you got so that's the phosphate backbone. So that's our guanosine, cytosine, thymine, and adenine. These are hydrogen bonds. Between adenine and thymine is 2 hydrogen bonds. Between cytosine and guanine is 3 hydrogen bonds. Phosphate backbone. There's oxygen between CH2 and the phosphate. Oxygen between the CH2 and the phosphate. Okay. This hydrogen is coming off at every point. Okay. Oxygen to hydrogen bonds, nitrogen to hydrogen bonding. Okay. I think I have completed it. So I believe there's over 100 atoms in this. You got I think it basically continues onwards. So that's 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So when people talk about 5 prime and 3 prime ends of DNA, RNA, they're referring to these carbons. It's just a carbon atom. It's just like the first, second, third, fourth, fifth, but we call it prime instead. So the 3 prime end, that's 5 prime to 3 prime. This is 5 prime to 3 prime going the other way. 5 to 3, 3 to 5. Okay. So it's just the way of ending the molecule. At the 5 prime carbon, you get the new phosphate backbone attached. And at 3 prime carbon you get oxygen leading on to the phosphate in the background. So that's where that comes from. I think there's over 100 atoms in this. I think it counted at about 128 atoms. The difficulty is not drawing it, the difficulty is fitting it into paper. So if I turn it over, again, you've got phosphate, oxygen, CH2 attached to the 5 prime, and coming off the 3 prime oxygen, phosphate, oxygen, CH2 attached to the 5 prime, and so on. The structure of RNA, let's see. so this is in coronavirus. Okay, in coronavirus, and you have so that's adenine. This is uracil. Okay. I want to point out to you when I got the bond kink there, the bend, that is not. It is not a nitrogen to carbon. It is a straight bond. Sometimes you have to draw bonds with little bends, but it's not. That's just basically a carbon to nitrogen bond like that, carbon to nitrogen. Okay, it's the same here, carbon to nitrogen. I've had to bend it in order to fit the molecule in. Probably really the molecule would be a little more sort of bent around or something, but I'm trying to just illustrate it to you. So this is RNA in coronavirus. That's riboneucleic. Says okay, RNA, which is ribonucleic acid. So yeah, so that's as I said, not a C there. Okay, it's just there's no C in the middle. So that's the and also in this H, this is in uracil, but this is in thymine, that is equal to CH3. Okay, so that gets replaced with CH3 in thymine. Okay, so that's the uracil molecule. Okay. Yeah. That's uracil molecule. I think there's everything there. It's obviously it repeats the phosphate backbone, just goes off. In humans there's 3 billion each. Well, it's not 3 billion actually, it's RNA. In coronavirus it's I can't remember how many bases, how many nucleotides there are, but it's several thousand nucleotides long, and you got uracil, adenine, cytosines, guanines all the way along. Sorry, cytosine, guanosines all the way along. UAGCs all the way along, forming the various genes and gaps. Okay, so don't think I missed any hydrogen. I've got everything. Double bond, carbon, hydrogen, carbon, hydrogen, hydrogens, oxygen there, oxygen, phosphate, oxygen, CH2, oxygen, phosphate. Yeah, I think that's it. Okay, so that's basic structure of uracil. Okay, in previous video I drew out the structures and it was a bit of a mess. It came, although I noticed, sorry, the first video was a mess. The second one, I noticed there was a double bond missing in the hydrogen in the wrong place. So I'm redrawing it. There's also, I missed some hydrogens and carbons out, which isn't actually such a problem. I'm drawing this out to demonstrate what I mean. Normally you can draw a molecule like this. Another way of doing it, often when you're drawing fat molecules, you'll do a CH3. So each of these vertices actually is a carbon, and you assume that there are hydrogens coming off. Okay, so when you draw this, it should actually be drawn more like this, because each sort of like that. When I draw things like that, I was assuming there's a carbon there, okay? Or I assumed there was, if I'd drawn that as a single bond, I'd assumed there was a hydrogen there. But I'd actually missed a double bond out in the structure and put a hydrogen in the wrong place, and those other hydrogen carbons missing. So I thought I'd do the whole thing again carefully